Hello everyone! We continue to study physiology, and today we have practice 6, physiology of nerve cell and nerve fiber. In general, physiology of the nerve system. Let's start studying it from the periphery, and today our lesson will be devoted specifically to nerves and nerve fibers. But today is the first lesson on the study of the nerve system, therefore we repeat the lecture and it will be an introduction to the study of nerve system in general. In the practical book today we have 3 lab work and we have videos for these lab works, and in addition there are a lot of videos on YouTube. As usual, I will leave the links in description. Okay, let's start. Well, let's move to this slide that explains us what is nerve system. It is a system of organs and structures that carry out the regulation of all life processes in a body, integration, coordination of the activities of all organ and organ systems, ensure interaction, traffic, adaptation, communication, and all general process inside of the body. Especially we have communication with the external environment and formation of non-direct behavior for animals. The body responds to irritation with a reflex, and this way nerve system can control all options. The nervous system can be divided in 2 major regions, the central nerve system and peripheral nerve system. The central nerve system consists from the brain and spinal cord, and a peripheral nerve system, its motor part, somatic part, and automatic part of nerve system, vegetative nerve system. All these parts have the peripheral part, and especially for today we have practice about nerve fibers. So we will divide these nerve fibers depend on the nerve part of nerve system, somatic and vegetative nerves. I hope you know what is this nerve. A nerve is a collection of nerve fibers. A nerve fiber is a part of a neuron, but today we will discuss about peripheral somatic nervous system, namely nerves. But first, let's see how the central nerve system is built, since nerves do not come from anywhere. They come out of the central organs, and so that we do not mention them further, their cells will be considered briefly right now. It includes nerve cells, neurons. You see the body of neurons, dendrites, it is a kind of fibers, and one very long, usually very long axon, and a lot of different cells, the cells which do their function, and these cells has a name, neuroglial cells, neuroglia cells, and briefly right now about these glial cells. It can be central glia or peripheral glial cells, support neurons. They don't generate impulses, and we will consider all these cells on 4 types of such cells. Central glia is divided into macroglia and microglia. Macroglia are oligodendrocytes, astrocytes, and epidermal cells. Peripheral glia are Schwann cells and satellite cells. On this slide we can't see the Schwann cells because it is a part of central nerve system, brain, but later we will discuss about these cells. Let's start from the astrocytes. You see, what do astrocytes glial cells do? The major function of astrocytes is to maintain and in a variety of all ways an appropriate chemical environment for neuronal cells. They have a regulatory role of brain functions that are implicated in neurogenesis and synaptogenesis, controlling blood-brain barrier permeability and maintaining extracellular homeostasis. And as usual, I will leave some links about all glial cells, and you can find this in this video information about astrocytes. Why this name? Because of it is like a flower, Astra. Even in this video you can find new functions for astrocytes, and these cells are not as simple as they seem at first. As usual, I will leave links for this. Especially about astrocytes, the second video. Next cells from glia, it's oligodendrocytes, which are also in the central nerve system, lie down eliminated lipid-rich wrappings called myelin around some fibers, but not all. Especially around axons. Microglial cells, next one, brain development primarily through two routes: the release of diffusible factors and phagocytosis. Macroglia phagocytosis, many products in the brain, including synaptic elements, died cells, even sometimes living cells, sometimes part of cells. So these cells removed wastes from the brain, from the spinal cord. Next, glial cells, ependymal cells. How you see, help circulate cerebrospinal fluid. Not only this liquid in ventricles of brain, the liquid is too, and these cells are inside these cavities. These ependymal cells are ciliated epithelial glial cells that develop from the radial glia along the surface of the ventricles of the brain and the spinal cord. They play a critical role in cerebrospinal fluid production and homeostasis, brain metabolism, and the clearance of waste from the brain. Ependymal cells are covered with short non-motile progenitors called microvilli. Here it is better, we can see. This increases the surface of area and the ependymal cells and allows them to reabsorb this liquid and waste products and something on. Next one. Now there are two types of peripheral glial cells on the slide. You see satellite cells and Schwann cells. These are satellite. Here you see little cells. In the peripheral nerve system, Schwann cells, the second name neurolemocytes, perform functions similar to oligodendrocytes from central nerve system, and they are myelin cover of nerve fibers, and these fibers is thicker and their functions are different from unmyelinated fibers. Here you see the Schwann cell around the axon, and function we will discuss a little bit in continuum. Here we see the difference where we can find the oligodendrocytes in the central nerve system here, and how it built it, and Schwann cells on the periphery, neurolemocytes, the second name. Here you see how the satellite glial cells surround a sensory and autonomic ganglia. Ganglia are realized stations where one nerve enters and another excites. In autonomic involuntary nerve system pathways, satellite glial cells respond to chemical messengers, neurotransmitters, and optimize them so that vital responses such as heart rate and vasoconstriction go as smoothly as possible. In the sensory nerve system, satellite cells regulate potassium levels, and the neurons respond to peripheral without the presence of neurotransmitters. So next slide, I want to show two more types of glia. These are recent scientific discoveries. Radial glia. This type of glia we know a long time and only now found in specific areas of the central nerve system. This subgroup includes the Bergman and Muller cells of the cerebellum and retina in the eye. Radial cells modulate neurotransmission and optimize how information is processed. By forming a framework or scaffold of which other neurons can travel, radial glial cells are highly communicative. They also play roles in ion homeostasis, increased synapses stability, and improved brain plasticity and neuroprojectants. This is done by regulating the surrounding extracellular fluid. And one more, enteric glia are found in the intestines. They assist with good motility, peristaltics, and enable contact between different cells of the intestinal wall. The enteric nerve system, or special system inside of these tubes, is secondary in cells population only to the central nerve system. Enteric glial cells seem to fulfill the characteristic of the other nervous tissue cells, such as astrocytes and oligodendrocytes. The fact that these cells are more broadly differentiated than any other types of glial cells makes them interesting subject. That is already known is that electric glial cells, enteric glial cells change in function and from according to the location, the age of the host, even the person gender. So it's very interesting to know how works these parts of glial cells for animals. Maybe it is in the future, it's a scientific development. So next slide. And so we move on to studying the structure of the neuron. I hope you know all this information from the histology and even from biology, from school program, but I leave a link to this information, this biology dictionary, very interesting information about neurons. The parts of any neuron are the soma. Also how long these parts the soma can be different shapes, and usually neuron has a lot of fibers, dendrites, and one axon. We can safely say that all neuron structure consists of a cell body, axon, dendrites, axon hillock, it is a special place, and terminals. A neuron is a single nervous system cell that receives, processes, and transmits electrochemical messages from and to other cells. Neurons connect different areas of the central and peripheral nervous system, stimulated at one end by electrical or neurotransmitter activity. A change in membrane charge is initiated and sent as an electrical pulse, action potential, along the length of the nerve cells. This releases neurotransmitters and at the end of the neuron that stimulate and response in the next cell. Neurons fall under various categories. However, all types of neurons function as electrochemical messenger cells. Now here you see the contact with the neuron and muscle tissues by axon, neuromuscular junction. You know this information from the previous practical class, and if not, you just watch this video. Next slide about structural classification of neurons. Neuron categories or neuron types can be split into 5 groups. Unipolar, how you see, bipolar, it is better to watch on next slide. Unipolar neurons, bipolar neurons. Here you see only 2 fibers, one dendrite and one axon. Usually we can see the cells in a retina. And next one, it's multipolar neurons. You see almost all nerve system consists from this type of neurons. Functional classification of neurons. Next, sensory neurons. We can call that neurons afferent neurons. Then interneuron and motor neuron or efferent neuron. So here we see the reflector arc. The primary neuron classification method distinguishes between central nervous system neurons and peripheral nerve system neurons. This category simply describes the location as within the brain and spinal cord or outside if the brain and spinal cord Neuron function remains as always the same and you see in central nerve system we have some neurons, and on peripheral part of the nerve system we have nerve cells too. Next slide. It's where we is now the peripheral nerve system, and all this practice about somatic nerve system. And we now will start to study cranial nerves and then spinal nerves. And some weeks later we start to study autonomic nerve system. So sensory neurons are primarily unipolar and found in both peripheral and central nerve systems. They pick up information according to the 5 senses: smell, taste, hearing, sight and touch, as well as data that helps us to know how our body is in position and in a balance. From the proprioceptors, for example. Spinal motor neurons are also part of the central nerve system. Think of the knee-jerk reflex, or how the muscles of our intestines work harder when you are resting. Upper motor neurons travel between the brain and spinal cord, and lower motor neurons between the spinal cord and muscle. And lower motor neuron always end at muscle fibers, and it's predominantly multipolar. Interneurons are rarely neurons connect sensory nerve cells to motor nerve cells, as shown on the previous slide diagram. There can be more than one interneuron in a body, and here we see the somatic nerve system, also known as the voluntary nerve system, is a part of peripheral nerve system. It consists of neurons that are associated with skeletal or striated muscle fibers and influence voluntary movements of the body. So let's start from the cranial nerves. Cranial nerves are motor, sensitive or mixed neurons that bring motor and sensory messages to and from the face, neck, and even shoulders, as well as many internal organs such as the heart, lungs, and gastrointestinal tract. I make with different colors. The cranial nerves are 12 pairs of nerves which arise from the brain and have various motor, sensory, special sensory, and autonomic function. For clarity, a special sensory function is any sense other than touch, specifically vision, taste, smell, hearing, and balance. And next slide, here we see 12 cranial nerves and their function as follows. You can watch what kind, where this nerve start from the central nerve system and what function it do. Now consider the spinal nerves. Next slide, you see spinal nerve topography. This all spinal cord segments in some common species. As you know, they originate from the spinal cord, and the spinal cord is located in the spinal canal of the spine. A segment, as you see, a lot of segments for all base. You see cervical, part, lumbar part, conus, and for animals we have different numbers of these segments. On this slide I found the spinal cord segments in some common species. You see dog, cat, cow, horse, as you know from a course of anatomy. A segment of the spinal cord is located in vertebra. Soon we will study the spinal cord, but for now we are only interested in processes, roots. Each segment has its own structure, and we will discuss it now. And we can find this slide and the picture of ganglion. Here you see in the dorsal root of spinal nerve or posterior root of spinal nerve or sensory root is one of two roots which made from the spinal cord. It emerges directly from the spinal cord and travels to the dorsal root ganglion. This part here and here you see, and on this picture here yellow color. Nerve fiber with the ventral root when combined to form a spinal nerve. All roots together combine and form the one nerve. The dorsal root transmits sensory information, forming the efferent sensory root of a spinal nerve, and ventral root move from the gray matter of spinal cord motor information, efferent information to the effector. Different types of effector we can find, and there you see on this slide the part of dorsal root ganglion. You see a lot of fibers from it. Here this spinal ganglion we can find on the dorsal root, and as you see one spinal nerve consists from the different fibers. Sensitive information move to the central nerve system, and this fiber is blue, and this information, afferent information, move in spinal cord. From the spinal cord information from motor neurons move on this way. Here we see difference in part of vegetative autonomic nerve system, but usually move in a spinal nerve motor information to the effector, for example just muscle. And as usual I will leave the links to the video about cross anatomy of the spinal cord and spinal nerves. Here you can find more information. And next one, why is this information so important especially for vet doctors? It is because we in future on a surgery we will do the blockades, anesthesia of these nerves. For this usually we use special liquids, for example lidocaine, novocaine, and here you see blue area where we use this liquid, and even you see the doctor move, do injection, use special equipment, and here on the bones you see where these cones, dorsal cones, sensitive cones, they are blocked in this place. That's why it is very important to know where is the area where you do some operation, for example, and what part of nerve roots move through the body, what part, myotomes, Metamers is a part of the body connected by innervation with certain segments of the spinal cord. What is the metameric segmentation of the animal body and human body? It is the division of an animal's body into linear series of similar segments, tetramerers, by the repetition of organs and tissues at intervals along its body. The same metamers, but we can find the same metameric segmentation. It's in enteric, mesodermal phenomenon. The basic segmental divisions are the body musculature and the cellum. The same. This division is very important to know for performing local anesthesia, how I said, Novocaine blockades. And here you see the map of dermametamers and myotomes of the human body, and the same is the animals too. And for visceral, how you see organs, there is similar. And even damage to the spinal cord segment in this case leads to the disorders of function in areas of the body associated with it. So now let's understand how the nervous system controls the function of the body. Reflex, where is it? What is a reflex? Reflex is a rapid automatic response to the specific stimulus. In its simplest form, a reflex is viewed as a function of an idealized mechanism called the reflex arc. You see, it's irritation from the skin, and irritation of receptors moves the signals through the nerves into the perimeter, and then from the ventral root to the effector. There are three common somatic reflexes discussed in the most anatomy and physiology courses: stretch reflex, withdrawal reflex, and cross and tensor reflex. And reflexes arcs are somatic and vegetative. As you see, it is a difference. It is left part, it is a somatic reflectoric arc and vegetative reflectoric arc. But when we will study the autonomic nerve system, we will discuss about all these parts of this nerve arcs. Now consider the example of the somatic reflex arc. We have the receptor, for example, on the skin, detects the stimulus. It's the receptor end of a particular dendrite or a specialized receptor cell in a sensory organ, for example, hair, eye. The second sensory neuron afferent, here we see the green line, and this nerve impulse move through the body to the spinal cord, in this case to the ganglion, where in this ganglion we can find sensitive neuron, afferent neuron, and this neuron transmit the signal to the interneuron here, but this is a recurrent components, not necessary components. Sometimes some reflector arc don't have this interneuron. And next one, it's a cell body of afferent neuron or motor neuron that transmits a signal to the muscle. Transmit the signals blue line, it is a nerve fiber, afferent neurofiber to the muscle. Muscle, usually we say that it is the effector. These muscle fibers or effectors which perform the reflex actions. That's this hand just move, and in most cases, however, the basic physiological mechanism behind the reflex is more complicated than the reflex arc theory would suggest. Additional nerve cells capable of communicating with other parts of the body beyond the receptor or effector are present in reflex circuits. As a result of the integrative action of the nerve system in higher organism, behavior is more than the simple sum their reflexes. We will discuss this later. So, next one, here we see the monosynaptic and polysynaptic reflex arc. It depends how much neurons we have in these parts. We have interneuron or don't have. And next important, we see that these green and blue fibers are part of the same nerve. Somatic nerve fibers are covered with myelin, as you know, and what we have here, we have two fibers. One, it's a sensitive fiber, and then we have motor fiber. And how it visible in a nerve, we see a lot of fibers inside the nerve. Part of a nerve covered with several layers of connective tissues. Here we can see better, and a lot of segments inside, and blood vessels, fascias, and even we see axon with the myelin sheets. A part of nerve on this slide, the reds. We see right celiac nerve looks natural. You see it's natural photo, and even green color, it is the myelin. Myelin, it is almost fat, just fat, very fat, inside of a lot of fat inside of Schwann cells. Usually a lot of axons covered with these cells, but not all. Not all fibers are myelinate with myelin, not covered with Schwann cells. In the nerve sheets there are also unmyelinated nerve fibers. Usually these are dendrites. Myelin is highly resistant. It performs the isolating function, and you see the gap between the two segments, Schwann cells, is called the node of Ranvier. Maybe next slide it is better. You see this is the body of Schwann cells, and it is node of Ranvier. This place is of great importance in carrying out the action potential in nerve fiber. But first let's learn about the potential in the unmyelinated fiber. You can watch the practice about formation of action potential. It is the second practical class, but I hope you know this, how action potential move through the nerve, features of signals conduction by unmyelinated fibers. At first, signal does sequence, and all fibers at once is seized with excitation. Signal goes with small rate, and signal goes with discriminant. So without isolation of Schwann cells, sometimes we just lost the signal, this action potential, and you see how it works. Very low speed, very small rate of moving the signal or this electricity on the surface of the nerve fiber. It is about 2 meters per second. It is very little. But if we take the myelinated nerve, myelinic fibers, the signal persists in skips and all fibers at once, is not sized by excitation. Signals goes with the high speed. It is about 120 meters per second, and signal goes without discriminant. And here we can find the video about saltatory conduction and conduction through myelinated nerve fiber. And it's very interesting how the signal moves through this nerve. It can to jump through the Schwann cells in place not of NVA, and thanks that the transmission of the signal can move very quickly, so quickly that it 100 meters and even more meters through the body. So we have some laws of conduction, of stimulation along nerve, and it is special laws we will discuss now. It's like special rules, so-called laws. At first, two-way conduction of stimulation in nerve during the bringing of the threshold of threshold stimulus on the nerve. The stimulation will spread in both sides. Bilateral conduction, but only through the synapse. The synapse, it is a special place which can't move. Just the signal don't be able to move after synapse. Isolated conduction because of Schwann cells and anatomical and physiological continuity of the nerve, not damaged, especially not damaged anatomical, any injury of nerve, and physiological. A phasic reaction of living tissues to a stimulus of specified duration and states by which the tissue's excitability, conductivity, and normal process of excitation are temporarily suppressed. The phenomenon of parabiosis was demonstrated, and a theory was developed by Vedensky on the first years of 20th century using the neuromuscular system of the frog. The use of such drugs as novocaine introduced the nerve into a parabiotic state. It is the next slide about physiological uninjured nerve fiber. What is this? If we just cut nerve, it is this nerve can transmit the signal. We understand this because just two parts of nerve don't contact with each other. But what happens if the nerve isn't damaged but this nerve can transmit the signal. This state have some, you see a parabiotic state, and each state has some phases, leveling phases, paradoxal phases, breakdowns phases. And one more video that shows this laboratory work. Also, we don't have such in the workbook, but this laboratory work illustrates the laws of conduction and excitation along a nerve fiber, and then we will see all these lab works, and about parabiosis too. So it's the first work for today. We see it is the work about definition of throughput or irritability of nerve and muscle. Previous our practical work was about muscle fibers, and now we discussed about nerve fibers, and now just know which of these tissues are more excitability or irritability. So let's watch the video. Here we see we have the muscle and we have a nerve, and we use special equipment. It is electrostimulator with the electricity, and with the electrodes we can just touch nerve or muscle cells. And what happened in this case? You see, it is a contraction of the muscle. Usually we need more high electricity signal for activation of muscle fibers, for generation of action potential. So here we found the real buzzer.
Мы определяем порог возбуждения для мышечной ткани. В данном случае порогом возбуждения для мышечной ткани является 0,3 вольт.
So, like a conclusion, in this work we said that nerve tissues are more irritable than muscle tissues, and you see here it's a new work about physiological integrity of the nerve for conducting excitation. And it's just description of this lab works. All description you can find in our practical And here we see all states of parabiosis according to Vedinsky, and even description how we continue this lab work. Previously we prepared the frog, as you can find in your practical book description, and then just touch the stimulator to the nerves and watch the effect. Then after that, if all nerves works, you see the contraction of muscle. We see so this nerve can transmit the signal. Celiac nerve here, and after that we can take the ammonia or iodine solution, and it is also possible to carry out the ligatura itself. You know, the nerves is never ligatured if we don't want to damage it. Well, in this case, I see that the harmonical damage was carried out and ligatura was applied. And after action of ammonia, let's watch 10 minutes just it takes. And what happened? With ammonia this irreversibly, but if we use the Novocaine solution after action of this liquid and after some time, about half an hour, it's the function of nerve and nerve be able to move and transmit the signal, reversibility. So we have the next practical, what we'll find. Its next work, isolated conduction of excitation along the nerve fiber. So in this video we have two, and let's watch the video to this lab work. About laws of transmitters of signal. Here we see three parts of the nerve, some fibers of the celiac nerves, and each roots of this nerve innervate different muscles. And just watch on the reaction of the different muscles if we touch one from three nerve spots. Here you see this nerve fiber innervate only muscles of the hips. And the next law about anatomical uninjured, and why it is very important. If we just cut the nerve, the signal can't move through this tissues, through this nerve to the tissues. And here we just cut the part of the nerve. And here we see that we put the ligatura on the nerve, and this nerve are damaged, or fibers are damaged, and the signal can't move easily to the muscles. And the next law, it's bipolar transmitters of signal. The signal can move only to the synapse, move in two sides, but only to the special place, synapse. So it is almost all work for today, and in the next slide let's see this slide. Let's watch some your homework on our Moodle page. You should watch the video about saltatory conduction of signal in a nerve, action potential in a nerve, and nerve impulse, all molecular mechanism. Also, you have the next test. It's the text physiology of nerve fiber, and in your practical, you should read the text to the lab works, and in your book Animal Physiology from Gens to Organism, you should read the pages about nerve system. See you later. Bye bye.